So on the quiz, it mentioned that tDNA is integrated, single-stranded, but on the PowerPoint, it mentioned that double-strand synthesis occurs first and then it's integrated. So what happens first? Is it integrated as a single strand or as a double strand?
Okay, the question was, if this is tDNA.
Yes.
tDNA travels from Agrobacterium to the plant as a single strand. It's coded with VIR-E2, and it has attached to it covalently VIR-D2, which is the endonuclease. And when it goes to the nucleus, it is then duplicated, and it's thought that the double strand form is what is integrated, no matter what it says on the slide.
Okay.
It's just like bacterial conjugation. The second strand is made in the recipient cell, and then it's integrated by recombination. Here, the second strand is made in the recipient plant cell, and it's integrated by an unknown mechanism, probably, again, recombination, but non-homologous recombination. This thing is still too loud. Okay. Another question, general question. You want to make sure I cover.
SHEV-E and overdrive. You mentioned once that SHEV-E has to do something with overdrive, but when I was studying, VIR-C only binds to overdrive, and SHEV-E binds to outer surface of.
Yeah, you're the second person to draw my attention to that. I probably misspoke in the lecture. I was so enthralled about the little joke between SHEV-E and overdrive. So I'll show in a minute, I'll show the agrobacterium model, and we'll talk about the role of SHEV-E and arabinose. But you're right, VIR-C binds overdrive, not SHEV-E. SHEV-E is chromosomal. CH stands for chromosome, V for virus. So SHEV, it happens to be gene E. So it's a chromosomal gene that codes for a protein that binds arabinose, that in turn interacts with this very plasmid domain of VIR-A. Easy to show it with a diagram. Does anybody know the consequence of SHEV-E protein having bound arabinose? What happens, what's the consequence of it now binding the periplasmic domain of VIR-A?
Increases sensitivity to
acetylserine. Right. It increases by about 100-fold the sensitivity of VIR-A to detect acetylserine. How is acetylserine detected? At what part of the molecule? It's a little hydrophobic. That's a clue. Membranes are hydrophobic. They actually dissolve. Yeah.
Inside the cell.
Yeah, I said inside the cell, but I've done more reading on it, and they haven't actually mapped the domain. They know it's not in the periplasm. They think that it's inside, but other people say it's in the transmembrane domain. It may actually interact with VIR-A while it's dissolved in the membrane itself. So I certainly won't have an exam question that will ask you to distinguish between those possibilities. Any other general questions before I?
Dr. Gurley, we have a question in the distant sphere.
Okay.
Wendy would like to know, in the audio file, the PowerPoints for cell replication had a change from compatibility to incompatibility. Which is the correct term, and does incompatibility groups relate to CoE1?
So if you've seen the lecture, you know that that's the one thing that continues to haunt me. For years, I've always said one when I mean the other. Incompatibility groups versus compatibility groups. It's an incompatibility group. An incompatibility group means they have the same origin. So it's a little bit counterintuitive. They are incompatible because their origins cross-regulate each other. So when it's trying to control copy number, let's say the maximum copy is about 20%, the origins are responding to the total copy number of the negative regulatory RNA, since that negative RNA can regulate either of the two origins. It's really now the sum of the two, and if it's the sum of the two, that has to be 20. That means you could have one of protein A and 19 of plasmid B. And so eventually, the one that replicates the fastest will win in a population. So they're not incompatible because they are at warfare, that they create toxins or they try to actively repress the other one. It's just a game of probability. After enough generations, you'll see that you'll dilute out the slower replicating plasmid because they're under the same copy number control mechanism. They're regulated by that same RNA1. Any other questions?
Can you quickly go over right border versus left border?
Right border versus left border. Okay, I'll do that when I get into the agrobacterium stuff. I don't know whether I completely answered the question that was coming from online. Incompatibility groups, there's another aspect of that question, and it's if you have two populations that start out with the same incompatibility group, which means they have the same origin, they cross-regulate each other, and you keep them separated for 500,000 years, it's entirely possible that they will mutate away from each other. They will continually recognize their own origin because as the upper strand or the primer RNA2 mutates through DNA repair, that means the opposing strand, which is RNA1, will have the counter mutation. So it will always be complementary. So it will always work. The strain that's mutating is always self-complementary. But it may eventually evolve away or mutate away so far from its original state that it can no longer cross-regulate the other previous member of the incompatibility group.
So that means that they can coexist then?
Then they can, yeah, because they are totally unaware of each other. They don't cross-regulate. So each now is counting this way, independent of the other one.
So is this statement correct? The same compatibility group, so plasmids in the same compatibility group are not able to coexist. That's not right.
That's
It's
defect in an aging brain. I should never say compatibility group. So I should have said incompatibility group. So let's switch the word and call it incompatibility. Could you read the question and just say incompatibility? I'm trying to purge compatibility from my head.
Plasmids in the same incompatibility group are not able to coexist in the same cell.
That is true. Ones in the same incompatibility group are incompatible because they are the same. They compete with each other in terms of that cutoff copy number. It's the total number of plasmids now. And eventually, through many generations, one will lose out. Okay. Now I've got sort of a general idea of where you are in terms of questions, so I'm going to open up quickly the agrobacterium lecture, and I'm going to go over the mobilization plasmid concept and some of the basics, because there might be a problem on the test with that, and I know you're all keenly interested in that topic.
Dr. Gurley, if also you could just expand on the importance of COLI1, that seems to be.
I didn't quite get it. What could you repeat the question?
They're interested in COE1.
Yeah, I'm going to go into that in detail as if I've never lectured on it, which I haven't this year. So I'm going to completely explain COLI1 and all its parts and how those magnificent parts could be exploited as questions. Let's see here. There's a thing. You'll also note that I have I've taken out the agrobacterium slides from the extra chromosomal replication, and I've consolidated them in the PowerPoint, and one PowerPoint devoted to agrobacterium. I have a, let's see, window. Okay, so let me, I need to do a Zoom thing here and make sure I'm sharing the screen. Let's see, share the screen. Okay. Kaylin, can you see now? Can you see the PowerPoint?
Yes, sir.
Okay, good. So this is what I did cover in class, and I'm going to go to an area that sometimes causes people have questions about. So this is the configuration of genes with respect to SHEV-E, and you see SHEV-E is located on the chromosome. Chromosome up in the top right there, and it makes a protein that, of course, interacts. It's exported, and this is binding. SHEV-E is binding arabinose. Why would it bind arabinose? We'll see in a minute. Then, of course, you have other chromosomal genes that affect virulence. These largely make polysaccharides, filaments, and things like that that help attach the agrobacterium to the wounded plant cell. Okay, so I'll just put up here quickly the scheme. Here you see two molecules that regulate. The triangular one is arabinose, and the, I guess that'll be the pentagon, is acetylcyringone. It interacts either just below the membrane or actually in the membrane because it's soluble in membranes. But most two-component systems interact with this periplasmic domain. This is a classic bacterial two-component system. It has a membrane-embedded receptor, and then that activates through phosphorylation a transcription factor that goes and binds the genes that are in that particular regulon. So in this case, the signal does not interact directly with the periplasmic domain. In fact, there are two signals. One of the signals we talked about, acetylcyringone, which is produced by wounded cells, interacts in this region. But SHEV-E binds arabinose, and when it binds it, it then interacts with the periplasmic domain. My question is, how come two signals? Why does agrobacterium want two signals before it's going to cut out its T strand and ship it or try to ship it into a plant? What's the significance of binding arabinose? It's easy what the significance of binding acetylcyringone is. It's a wounded cell, but can you remember why it's interested in a wounded cell? Is everybody? Yeah.
It's easier to penetrate.
Because why is it easier to penetrate a wounded cell?
The cell walls are thinner.
The cell walls are thinner because the cell is dividing and repairing the wound. And so in that cell division, at the initial stages, the cell wall is very thin, and that's the major barrier. So agrobacterium has two questions. First, are you wounded? Acetylcyringone. And the second is, hey, are you still alive? Are you regenerating a cell wall? Are you growing? Because it doesn't want to infect a cell that's dead. So these two signals work together. It knows it's wounded, and it knows that the cell is in the process of regenerating itself. A thin cell wall is perfect for injection of the tDNA. Okay. So a two-component system, VIR-A receptor, VIR-G, the transcription factor, is activated by this transferase reaction, phosphotransfer, that now unmasks the DNA binding domain. So this now can go and bind to the VIR-Box promoters in the VIR region. See if we can get the light down here. Better. And here you see that happening. Which components of the TRA region are analogous to VIR D1, D2? This constant comparison of TRA and VIR, something you should have in your head. So let's, everybody understands that, right? The TRA is for bacterial conjugation. Through evolution, an additional copy was arose, and it became a substrate for experimentation. It adapted to transfer, using the same mechanisms, transfer a portion of the TI plasmid from agrobacterium into a plant, using the same strategy and the same machinery, essentially. So in VIR D1, VIR D2, what do they do in the VIR region? Anybody, somebody blurt out. What do they recognize the ORT, or what do they do? Yeah, the VIR D1 recognizes what is equivalent to ORT if it was conjugation, but this is virulent, so it recognizes the right border and cuts. It also recognizes the left border and cuts. I'm sorry. It recognizes the border, it does not cut. VIR D2 does the cutting. VIR D1 recognizes the right border, recognizes the left border, and does not cut. It's too stupid. VIR D2 is like master blaster. Master and blaster. D2 does the cutting. So that's analogous to Y and I, right? Y, TRA, Y being D1, TRA, I being D2, the cover. So be aware of that analogy. So the other analogy is the borders, right and left border. What are they analogous to in TRA or ET, the site of cutting? Why does it cut at right border first?
So the tDNA is transferred because it goes away from it into the.
That's the rationale. So it would start at the right and go to the left and then cut again. If it started at the left border and went to the right, it would transfer the entire TI plasmid, everything except the tDNA, because it would cut again and everything would be transferred except tDNA. So what makes it cut at the right border first? Overdrive. There's a sequence, a binding site for VIR-C. VIR-C binds right next to the right border, and VIR-C has affinity for this VIR-D1, VIR-D2. So on the exam, I believe there's a fill in the blank. I have more than a belief. I have certain knowledge. I'm going to ask you to fill in the names of some of these VIR proteins. And for some of these, the number is important also, like VIR-C doesn't have a number. VIR-D, though, does have a number, and they have different functions. VIR-D1, VIR-D2. So remember, D2 is the one that does the cutting. D2 is like TRA-I. VIR-D1 recognizes the border, like TRA-Y recognizes border T. There's also another one, VEER-E. VEER-E2 is the only one we talked about. What is VEER-E, somebody?
It coats it single-stranded.
A single strand binding protein that specifically binds the T region, the T strand. And it turns out you don't make much of it. You make enough to coat about one strand. So you don't get strand after strand after strand going into the recipient plant. Although in many cases you get more than one copy of tDNA integrated, and that's probably because more than one agrobacterium has latched on to that poor wounded cell.
Is that the protein that's comparable to protein A?
No, I don't know what the analogous protein would be. I don't think there really is in the TRA region. TRA uses just normal single-strand binding protein. But this is a specialty protein not used in normal replication, just induced to coat the T region. There, E2.
Wasn't there a quiz question comparing one of these proteins with protein A and also TRA-D?
TRA-D, there would be, I don't know about protein A. Protein A makes a sex pilus, and I don't know what the pilus
Oh
protein.
no, protein A and
D.
in rolling replication for, what is it, 5'? Maybe I'm confusing. Never mind. I'm confusing.
Yeah.
Okay, so just real quick back to the
Uh-huh.
so if the overdrive happens for transcription from the right to the left, from the right order to the left.
Just, is overdrive necessary for what?
Can you explain the overdrive just one more time?
Okay, maybe I have a, so right border, left border. So the first thing to happen is VEER-C binds to this overdrive sequence. VEER-C has affinity for this VEER-D1, VEER-D2 complex. So that's now two reasons VEER-D1, VEER-D2 has for binding right before left. It's an example of cooperativity. Strongly favored to remain stuck here first. In fact, this is probably pretty weak. The only reason it really binds here is because it's pretty much tethered. Replication starts and it can't go very far. It's just kind of forced to bind over here. So it binds here because VEER-C binds overdrive, and overdrive is located right next to right border. And that's why it gives VEER-D1, D2 two reasons to bind on the right border first. And notice this is a trick that Agrobacterium has learned. If it was a fertility plasmid, you would cut once and go all the way around the F plasmid, and then you cut again and make one plasmid. It would get ligated together, and it would be bacterial conjugation of a plasmid. But in this case, it put two cut sites, so it doesn't go all the way around. It thinks it's going all the way around, but it's only gone over to the left border and cuts. So you can imagine what I might do on exam, and I would call it critical thinking. But you can call it, I'm just messing with you, just having fun. But you can handle it. What would happen if I moved overdrive over here? What would be the consequence if I did that?
Affinity to the left border.
It cut here first.
Yeah.
And then we start transferring that way. And then we transfer the entire TI plasmid and it'd get over here and make the second cut. And it would transfer everything except the tDNA. Okay. The other is the concept of the binary of a vector. And what I'm going to do is mess with you again. I'm going to fiddle with it and ask you to predict consequences. So I might ask you, what is present on the mini TI versus what is present on the helper plasma? So you understand the concept of a binary vector, right? Two components are kept separated from each other. So unless you have both components in the same cell, mobilization will not occur. So you have to have the TRA, that makes the line, the channel, and all that stuff. And you have to have a site to cut, a site to nick, to start the process of transfer. They don't have to be on the same plasmid. So that realization allowed the development of two kinds of binary systems. One involves the mini TI, cloning it and modifying it in E. coli, and then transporting it into agrobacteria. That involves a binary system. And the other binary system is once it gets in agrobacterium, this little mini synthetic vector here is part of a second binary system. Again, it's the mobilization portion, but now the helper plasmid, which is designed not to facilitate transfer between two bacterial cells, but to facilitate transfer now into the plant. The helper role is performed by what in that second binary process? So you've got the mini TI. In the first transfer system, what you needed was a basis of mobility. You needed the ORI T to be on the mobility plasma. Let's go to another. What did I do here? So I'm going to go to this slide here. Here you can sort of see all of it. So the first binary system is transferring from E. coli into agrobacteria. You have a helper plasmid and you have a mobilization plasmid. In this case, it's called the mini TI. What is the minimum thing it needs before it can be transferred? Just an ORI T, doesn't it? Just the site to be cut, because it's going to be acted upon by the TRA-Y and I that's produced by the TRA region that's located on the helper. So all it needs is a nick site for bacterial conjugation. So you can think of this mobilization plasmid as just having a collection of ORI Ts. The bomb site, which is on the exam called the MOB site, the mirror image of that, I guess, for mobilization. Bomb, basis of mobility, MOB, mobility. It's really functionally an ORI T, a cut site. It has exactly the same consensus sequence that is recognized by the TRA system. It also has other ORI Ts, right? Or functionally equivalent ORI Ts. The right and left border really function like ORI T, don't they? The cut site. But they can only be acted upon by the components of the Vir region, the D1, D2. So when the mobilization plasmid transfers into agrobacterium, now, in order to go to the plant, Vir D1, D2 act at the right border and then cut at the left border and ship into the plant this little synthetic tDNA. The modified TA now, modified TA, sorry, Kaylin. The modified TI plasmid has deleted as the T region deleted. It can never mobilize itself. It doesn't have it. It can only act in trans on mini TI that's now inside. So you see that it's the modified TI is playing the role of the helper plasmid, except for genome modification of plants. So let's look at this again. This is the original obsolete vector. I captured this in an intermediate stage of development. Notice it has its own ORI-T, and that same sequence, identical, is located also on the mini TI. It's just called basis of mobility, or the MOB site. It's the same sequence. In this case, what's going to happen when conjugation occurs? Both plasmids are going to get mobilized, right? So I like to mess with that a little bit on exams.
So is it going to be 3 plasmids in agrobacterium?
Yeah. You'll end up with the helper in there and the mobilization plasmid, which is the mini TI. Most of these helpers now have the ORI-T deleted in the helper. So now it remains behind, and this now provides much tighter biological control, right? Because now if Agrobacterium had this promiscuous helper plasmid in addition to the mini TI, it could cause transfer from Agrobacterium to Agrobacterium to Agrobacterium in the soil. And since it's a promiscuous plasmid, it can function in lots of different bacterial species. It could start transferring the mini TI to Rhizobium, all sorts of things that exist around roots. So this was a biological containment feature. Separate the 2 components needed for transfer. So one gets left behind, the helper remains behind in E. coli.
If the ORI-T is removed, then how does the helper plasmid function? How does it have a cut site?
Yeah, because it never has a cut site. How does it function? It still has an ORI-V. It remains in E. coli forever. It just can't infect new E. coli.
No, but I mean, how?
But all these things, the tri-Y, the tri-I, they just go wandering around in some existential land of, will I ever find happiness? The answer is, you have to whisper in its ear, no, you will not. Unless some graduate student transforms you with a mini TI, and then it's happy day.
So it just, so.
And everything works. You suck the pill out, everybody draws it, everybody's ready. Okay, go. And it's just one thing I'm missing. I'm missing a place to cut back, and it's around here somewhere, and it's not there.
Okay.
It's sort of sad.
On the mini TI, why is the arrow pointing from left to right, like between the?
That is okay. We didn't say what is on the mini TI. This is just historically, how have we developed it? And this is something that might be able to do. I think it's one of the homework questions. What do you need? What is actually on the mini TI that people use every day to modify plants? Well, it has an ORI-V, so it can replicate in E. coli. That ORI-V also allows it to replicate when it gets into Agrobacterium. It's just a plasmid, it needs to replicate. It has a basis of mobility site, or MOB site, which is functionally an ORI-T. And then it has right and left border, and everything in between is synthetic. It has a selectable marker. Quite often, it's kanamycin resistance. This allows you to select for plants on plates. It's just on growth media plates. The plants that are still green after about 4 weeks have been truly transformed. Their chloroplasts are resistant to kanamycin. Their kanamycin contains, you have a selectable marker, and you have a multiple cloning site so that you can clone things in your genes of interest. Herbicide resistance, insect resistance, improve the nutritional quality of a crop, drought resistance, all sorts of stuff. Yep.
Question. So which plasmid would you say is an example of a binary vector and a disarmed vector?
Okay, what is a disarmed vector? It's disarmed because it can't form a tumor. So it can't make opines, and it can't make plant hormones.
Mini-TI?
The mini-TDNA, but it has nothing in between. The natural tDNA, I think that's a homework question. What are the two major classes of genes that are found in the natural tDNA? You know, that is 23 kilobases, about 13 genes through there. They fall into two general categories.
Opene synthesis and plant hormones.
Opene synthesis, which are these conjugates of pyruvate and alpha ketoglutarate with basic amino acids. They can't be metabolized by most eukaryotes, and most bacteria can't metabolize it. But Agrobacterium can break it down and use it as a substrate, so it's perfect. And the other thing it has are hormone genes. So just think about it. Agrobacterium has genetically engineered a plant to form a collection of cells we call a tumor, to grow like crazy and pump out its favorite food. That's pretty cool if you're Agrobacterium. So as it's pumping out its favorite food, when it rains, these openes are just leaching out of the tumor, flowing down the plant stem, and providing a banquet for all the Agrobacterium left in the soil. So you have a few pioneering Agrobacterium that actually make the transfer. They are encapsulated in that little drop of rain with this little speck of gravel, which makes a little micro wound on the plant. They transfer the DNA, and then the plant does the rest. Grows like crazy and pumps out openes. What's the function of the modified TI plasmid? What does it provide? It provides the Vir functions, just like the helper plasmid in bacterial conjugation provides the Tra functions. And what's the strategy? Floral dip is the strategy used to actually get Agrobacterium in solution here, actually into the plant germline, into the floral meristem that's eventually going to make seeds. Any questions on Agrobacterium and the mobilization plasmid, the mini-TI? You'll have 4 questions total on Agrobacterium. You'll have questions that you'll have to fill in the blank on a VIR-D gene. I'll tell you what it does and you fill in the blank as to what it is. And remember that the number is important, like VIR-D1 versus VIR-D2. It may be one that doesn't have a number, like VIR-C has no number. The ones that have numbers are VIR-D1, VIR-D2, and E2. VIR-A doesn't have a number. What is the analogous protein to Tra-J? VIR-G, right. VIR-G in the Vir region is analogous to Tra-J. They're both positive acting transcription factors. Okay, let's go to Coli-1, and then I'll look over my copy of the exam and see whether I've left anything out.
Dr. Gurley.
Question somewhere?
Your TA.
Yeah, Dr. Gurley online.
Yes.
Just by any chance, I don't have my book with me, but I'm getting a question related to the book. Is there anywhere within the book specifically that the students could look to go into more depth on the topic?
No.
Okay.
You're hearing it from the guy who lived it. This is as deep and warped as you can get. the slides give you all the information.
Okay, that's what I said, but I just wanted to make sure.
Yeah. I mean, you really don't want any more, do you? Because I got lots. Okay, let's go through this. Why don't I just go through real quick and slow down when we get to Coley 1. We're talking about connections between cell cycles and replication. So, of course, once you start replication, you are committed to divide. So how long does it take to go through the whole process from when you start replicating your genome to the time you get to divide? 60 minutes. Because it has two subcomponents, right? It has a C and a D part. Doubling time, which is really, this is the D, it's really how long it takes to synthesize or to replicate the entire genome. That's the major work. It takes an hour to make the 4.2 million base pairs. Make sure they're all repaired and everything's good. And then, after you've done that, it takes an additional 20 minutes to form the septum, to make sure you've got partitioning of the genome into the 2 daughter cells. So it's D40 plus C. Nope, it's, I think that I got those initials wrong. It's not. Let me get the C. It's C plus D. Actually, I have it. I explained it completely wrong. C is for chromosomal duplication. That's the 40 minutes. C is 40 minutes. So please do not remember D as being. D is for division. That's the 20 minutes. 40 minutes, C. D for doubling, which is means make the septum. Boy, good thing there's the optional file. Then there's the question, inevitable question about how can E. coli, if it takes 60 minutes, C being 40, D being 20 equals 60, how can it possibly divide in 35 minutes? And the answer is, it begins to initiate DNA replication for the daughters to divide before it's even formed the daughters. So you have overlapping cycles of replication. And that's, here you see, in this example in the book, it takes 35 minutes before you get cell division. You have the origin, you have the first duplication or replication occurring, and then after about 10 minutes, you start again on each of the 2 daughter strands. This is what eventually will lead to division of your 2 daughter cells. And you start that a generation ahead. It's like your grandma establishing your trust fund or something. So that's a multi-fork chromosome is what it's called. So in this particular example, you would have multiple origins. And here you see the overlapping concept. 60 minutes, C plus D, equals 60 minutes, but you see it's begun the extra round of replication before it's actually divided. So really, the time that it takes to double is just the distance between the 2 septum formations. 25 minutes would be how long it would take to double. If you slid this back even further, make the overlap even greater, you could get that down to, I think, 18 minutes is about as tight as you can make it. Okay, the second concept. Bacteria, as they grow, they elongate. And so if you looked at bacteria, you could pretty much get a measure of how rapidly they're growing just by looking how large they are. When they get to 1.7 microns, as they get larger than that, when they get to that size, the cell is so large that the components that regulate initiation trigger a second round of initiation before the cell has started to divide. It swells up and it dilutes out a negative factor when it gets to 1.7. And now the second round of initiation begins even before this E. coli has divided. It's simply by size, you can tell how many initiations are occurring at the same time. So if it's less than 1.7, there's only one round of replication occurring. If it's in between 1.7 and twice 1.7, there are two rounds of replication occurring simultaneously. If it was between 3.4 and what's the next? 5.1, there are three rounds going simultaneously. There will be a question, and it won't take scratch paper for you to tell me how many rounds of replication are occurring. The answer is going to be 1, 2, or 3, I'll tell you. It's not going to be some big complicated number. Just interested in the concept. And then how is mass titrated? There are two theories that both might be correct. You could need the buildup of a positive factor. Well, you need DNA A, right? So DNA A sort of fits that model. When you get a high enough concentration of DNA A, it congregates at the origin and eventually ends up melting the origin, triggering initiation. There's also a model that says there's a negative acting factor and it has to be diluted out by the swelling in the growth. The data is consistent with that also, but we don't have any clear idea of what these negative things may be. How many replication origins are active in an E. coli that's 2.1 microns in size? Well, that's above 1.7, but below 3.4, so I'm going to go 2. Now the last concept before we get to coli 1. It turns out that the origin and the place where termination happens, which is all the way across that big genomic circle, they have distinct spatial locations within the cell. The termination site remains at what will later become the place where the septum forms. And the origins are kind of stuck to the membrane, and they, as the membrane expands, as the cell expands, they separate from each other further and further. And then the septum forms, and the last part to be released from each other is right there close to where termination happens. Okay. Let's look at the consequence of that. As you duplicate the genome, of course, you get multiple copies of the same DNA sequence. That's the basis for bacterial conjugation, the incorporation of those transferred genes into the chromosome. E. coli's homologous recombination system works overtime. Bacteria hate repeated sequences. They try to recombine them. And so, by accident, sometimes, as you, in the process of replicating that other copy of the genome, you will get a recombination event. Sometimes yes, sometimes no. If you do not get a recombination, then there's no problem in separating the two genomes from each other as the cell divides. No problem. But in other cases, you will get a recombination happening very close to the point where you're getting ready to separate stuff, because when the two replication forks fuse, There's some complicated recombination stuff that needs to go on there that we haven't talked about. But sometimes it allows this sort of accidental recombination by the host homologous recombination system to act upon the two newly replicated genomes. If that occurs, it will create what's called a co-integrant. Both genomes will be linked together in one great big bigenomic circle. The problem is, how do you get out of that? You recombine a second time, double recombination, and it undoes that damage. And that is conducted by a specific recombination machine called the Zur site specific recombination. This Zur site specific is just there to get rid of this specific problem, and it's not general recombination, it is site specific recombination. It will only recombine when it lines up two diff sites. That's the diff site. And so the Zur site specific recombination acts at the diff site. So I'm going, why do they call it Zur site? It's Zur site specific recombination. The hyphen is between site specific. So the Zur system acts at the diff site to resolve the co-integrant into separate genomes. Okay, everybody clear up to this point? It's not that complicated, just gotta learn the terminology. And
Dr. Gurley,
yeah,
if you could just briefly repeat, go over the 1.7 to 3.4 ratio.
okay.
Thank you.
As a cell grows, you dilute out positive or negative components, and as it grows, you increase things like DNA A. So you change the concentration of components in the cell that regulate initiation of DNA replication. And so there's a definite relationship between the mass of the cell, which is strongly correlated with its length. And we just found empirically that when a cell is growing and gets to when E. coli is growing, gets to a size of 1.7 microns in length, it's a transition point where if it gets any larger than that, you can pretty much be assured that a second round of DNA replication has begun. So now you have this multi-fork situation in progress. Sometimes the cell is growing so fast that you will start DNA initiation 2 generations ahead of when the actual division will occur 60 minutes later. And so when a cell is between 1.7 and twice that, you have one additional round of replication occurring. 2 rounds, we'll say, happening. If it's between 3.7 and the next, which is 3 times 1.7, which would be 5.4, you're going to actually have 3 rounds of replication overlapping each other at the same time in the same cell before it actually divides. Is that clear to the people asking questions?
Yep, that helped. Also, one more question. They want to know about pictures or figures on the exam. Is that something they need to worry about?
Figures. Gee, the farm has to have some price. I don't know of detailed figures where I'll say label all these parts. There are figures that describe configurations of things on helper plasmids and stuff like that, but there's not any figure that I can remember that where you have to label all the parts of something. Like, for example, you're not going to have the TRA region and label all the genes. I'll describe the function of a trigene or the function of a Vir gene, and then you'll either fill in the blank or select it from a list in multiple choice. So you're not going to have to know the physical order of the genes in Vir or the physical order of genes in Tron. That's probably what they would really ask. Who knows? Okay, let's get to this topic that is the hallmark of excellence in medicine. If you don't know how COLI1 replicates, what kind of physician will you be? I jest. It's a very elegant molecular mechanism, so I include it every year, and it's in the textbook still, but we don't know how many of these origins work, but we do know a lot about how COLI1. COLI1 is a plasmid. It's just called COLI1. It's an evil plasmid. It makes a toxin. So when it infects E. coli, it brings along a poison. Why would it do that? Because also it likes to sell E. coli the antidote to the poison. I'm sorry, I bashed in your storefront. What you need is protection. This kind of stuff was invented long ago. So anyway, we know that COLI1 is has a copy number limit of about 20 copies per cell. So the question is, I mean, that's just enough for all your fingers and all your toes if you were counting. But E. coli, of course, and this plasma has no fingers or toes. How does it count? How could you build a counting mechanism at the molecular level? So this is really an illustration of sort of a machine, an algorithm. How do you count? How do you limit copy number? So these are the players. It's best just to describe it with pictures. So this is the origin right here. This is the part that has to melt open. This is where DNA polymerase has to grab a primer of some sort and initiate replication of the plasmid. This is ORI-V in COLI1. So it's all about regulating the primer in this case. You know, at ORI-C, it's all about regulating, recruiting the helicase. But here it's all about regulating the stability of the primer. See how that works. You have two promoters here. Promoter, or P, Roman numeral 2, and promoter P, Roman numeral 1. This promoter here makes an RNA that's about 550, 555 nucleotides long. That is the primer. It is way longer than you need to have theoretically for a primer, right? Because adenovirus only has one nucleotide, the deoxy-C. And yet this is 550 nucleotides of RNA that acts as a primer. It's RNA and it's long. Normally, RNA, of course, it's going to be made by RNA polymerase. This RNA is not made by primase. It's made by the regular RNA polymerase in E. coli. It looks like a messenger RNA in terms of its mechanism. So it's long, and RNA polymerase, as you know, tries to separate the RNA from the DNA, from the template. That's what RNA polymerases do. Yet, this particular RNA, you want to remain attached to the template so it can act as a primer. So how does that happen? How do you attach this long RNA, keep it stably attached to the template? The way it has solved that problem is to build in secondary structure, inverted repeats. So the single strands here are able to duplex with themselves. These complicated hairpins are thought to fold around the DNA and keep the RNA from floating away. That's not a very satisfying model intellectually, but it's all we've got. It turns out the formation of those hairpins is critical to DNA replication. So if you can control whether there are hairpins here in the 5' end of this primer called RNA2, promoter 2 makes RNA2, and RNA2 is the primer. This primer goes all the way through the origin, it turns out. It goes past the origin. It goes too far. So you make a primer, and it goes too far, so and the primer, though, doesn't go away because it has all this secondary structure here. So how do you fix the problem? You process the end. You cut the end of the primer so that it is exactly in the middle of the origin. And that involves the second component here. You have RNA2, the primer. It has secondary structure. It has to remain stable long enough for triple strand specific RNase H to process the end, which means cut it so it actually ends up in the middle of the origin. If you don't have these hairpins here, it's formed and just falls off, and RNase H can never cut it at the right place. What else is involved here? There's a negative component. So what is positive that facilitates replication? The primer and RNase H. Both of those have to be there to support replication. There are also two negative components to regulation. These work against replication. One is RNA1. It's in the same region as the 5' end of the primer. It's just going the opposite direction. So if you mutate here, in DNA repair, you're going to mutate the other strand. One strand is mutated, the other strand receives the complementary mutation. So these two RNAs, RNA2 and 1, can mutate for hundreds of years and still not disrupt anything as long as they don't mess with the hairpins. Because you mutate the top strand, the bottom strand, if it's a stable mutation, receives the complementary base change. So they always pair with each other through time. So you have RNA2 here. It's just made in the hairpin region. Now, what does that do? If RNA1 can form a duplex with RNA2, then it'll smooth out those hairpins. They'll just form linear double-stranded DNA. You smooth out, you iron out those hairpins, and the primer no longer stays in place long enough to get processed by the RNase H. So the buildup, the titration, the buildup of this inhibitor RNA, this antisense RNA, the buildup of this begins to shut down the effectiveness of the primer. And that's the counting mechanism. As you build up, RNA species 1 begins to eventually get to the point where there's so much of RNA 1 that it immediately hybridizes, gets rid of the secondary hairpin structure of the primer, which is RNA species 2. There's one more essential component. Let's look here in some of the slides. Now, this represents the hairpins that are formed in the first region. How can you hybridize a second RNA to that? Because, the second RNA is also going to be full of hairpins because it's complementary to it. These two RNAs are not going to want to open up and hybridize with each other. So it was a beautiful scheme except for one little sticky detail here. The duplexes don't readily open up and hybridize with each other. You need the assistance of a protein to open them up so they can find each other and form a hybrid. And that protein is called ROM. ROM straightens out transcript 1 and transcript 2 so they can anneal with each other. And now you see 1 is annealed at the 5' end of 2 with the assistance of ROM, and there's no secondary structure. Therefore, this RNA2 falls away from the origin. It cannot remain in place long enough to get processed or cut, so that the end now is in the middle of the origin. So, if you were, in a laboratory and you were trying to design plasmids and get expression levels and manipulate this, how could you increase the copy number? Several ways to do it. What would you do?
You could find a way for ROM not to be present.
Yeah, you could down-regulate the promoter that makes ROM. Down-regulate something that's negative. So you could do that, apply that same principle to P1. Mutate P1 so it's not as effective in making RNA species 1. Or you could make it so the production of the protein is reduced. You reduce the negative components, that means you're going to increase the copy number. You could do just the opposite manipulation to decrease the copy number. You could fiddle with the levels of the negative component, but you can see how you could easily also fiddle with the activity of the primer itself. You can make more or less primer directly. Whoa, I've given away critical thinking questions. Your head hurts, that's all that matters.
What's the difference between P Roman numeral 2 and RNA2?
P means promoter. So P2 is the promoter that directs the synthesis of RNA species 2, which is the primer.
Oh, okay. That makes sense.
P stands for promoter. Are there any questions on the regulation here?
You said you will give a mutation to the P1, but if once you give a mutation to the P1, and there will be a mutation to the P2, because there will be.
If there's a mutation in this region of the double-stranded DNA, one strand represents species 2, but it's DNA, but it's turned into RNA, and the other strand is an exact copy of the RNA species 1. So just in mutation, if you mutate one base, it'll either get corrected or it could be end up with a stable mutation, and in the repair process, it'll change the opposing base. So they'll always be complementary. So 1 can always regulate 2, even if they mutate, because they're mutating together in complementary fashion.
I know that, but you said if you want to get less copy of the plasmid, you will
mutate that. Okay, you mutate the promoter. Promoter is where RNA polymerase binds and activates transcription. Just make it so it's not a good promoter anymore, or make it so it's really souped up and hot. But you're not really modifying the transcript itself, just the region immediately up in front of the transcript, the promoter. In this case, it would be down here. Looks like my battery's going. So the last aspect of this, how I can torture you on an exam, is a question about, you've got 2 cultures that are growing independently. They started out in the same incompatibility group. But after 1,000 years, they've been separated, and now they're joined back together by people taking collections throughout the world, and they put them in the same, they isolate the DNA, and they put the 2 plasmids into the same cell. And what you find is that the 2 now are not in the same incompatibility group, which means they have their independent counting system, right? If there were 20 copy plasmids in the old days, you'd get 20 of A plus B, because they cross-regulate each other. The negative RNA, the RNA species 1, it can shut down its own plasmid replication, or it can shut down plasmid B because they have the same origin. So the cell is really counting for a total of 20 origins, whether it's plasmid A or B. And so that puts them in direct competition with each other. But if they've been separated for a long time, they're each going to be sort of drifting their own way through mutations. And there's not going to be much selection here because these mutations, if they're just in a hairpin region, are not really coding for proteins. They just have to remain self-complementary and be able to form these hairpins, which is easy to do. So if you put them together after 1,000 years, you may find that now you have 20 of A and 20 of B. They don't see each other anymore. They would now be not be in the same incompatibility group because their two origins have diverged from each other so much that they don't recognize each other.
Do you have more than 2 plasmids in a single incompatibility group?
Yeah, you can have, as any place you had the CoLE1 origin, or something very, very similar, you could have it on 10 different plasmids, and then you put them all in the same cell. You still can't give it 20 copies total of the whole bunch.
Excuse me, Dr. Gurley.
Yep.
Is it possible that you could just rotate the camera a little bit so the students could see the lecture screen?
They can't see the lecture screen?
No.
I'm sharing it.
No, they can see it. They want to see you pointing, like where you're pointing.
Pointing.
Yes.
Oh, that's a problem, isn't it? Okay. So what part do you want me to repeat? I can point with the mouse over here.
Yeah.
Okay, let me do some of that mouse. You can see this pointing. So the top strand of the DNA here is the same as RNA species 2. It's just a copy of it. It's an RNA copy of this top strand of DNA. RNA species 1 is made by promoter 1, and here you see it in this kind of purple color. It's just a copy of the bottom strand. So if you pick up a mutation in this area in the top strand originally, by repair, it will repair it so that the bottom strand is also remains complementary to it. And so the two strands, as with all mutations, as you pick up mutations, the two strands always remain complementary to each other through a mistake in repair. You get a C changed to a T, then you'll find in replication that other strand now will become the C changed to T, become an A. The two strands will always remain self-complementary. So you can get an origin begin to pick up mutations and drift away from what its original sequence was. But still, it will not disrupt the function of this control. But if you put it now back in a plasmid that has the old way, because you kept it locked in a freezer at -80 for all those years, then you would find that this RNA1 down here would no longer be able to form a duplex with RNA2. And so they would not be able to cross-regulate each other. Now each plasmid could have a total of 20. Did that help the person?
I believe so, yes. In general, I think for, they're saying yes. In general, I think for these figures, they would like to.
Just they want me to continue that way.
Yeah, if possible.
So they through with figures. Okay.
Also, there's a question related to RNA and RNA-H for the CoLE1 replication.
Okay, RNase-H.
Yeah, they just want to know the function.
Screen RNase-H. This RNA transcript for RNA species 2 actually goes right on through the origin. And it has to be cut at a second step or process, so the 3' end corresponds with the middle of the origin. So it's not enough just to make the primer. It has to be processed or cut by triple strand specific, the 1, 2, 3, triple strand specific RNase H. An RNase H is one that cuts an RNA DNA hybrid. It just can't cut regular RNA. The RNA actually has to be paired with DNA before it can cut the RNA. And here you see the RNase H approaching this. So you need to have that RNA species 2 remain attached long enough for the end to be processed to form the correct primer. And that is dependent on these hairpin loops. So this antisense RNA made from promoter 1, it base pairs here in the 5' region and straightens out by forming a hybrid or a duplex, eliminating the hairpins, which decreases the stability. With the assistance of the ROM protein, ROM helps both of those hairpins straighten out and hybridize with each other instead of each strand with itself. Is that better? I'm hoping.
Yes, sir. Thank you.
Okay, I'm going to stop screen sharing and you're going to get just the classroom view here. See if I can figure out how to do that in my spectacle. It's got to be stop share somewhere. Stop share, there it is. Okay, notice UF is written backwards. That's magic. That means when they see me, I'm backwards. My hair is what's left is parted the other way in actuality. I think in the actual recording it gets flipped again. What we got here? Nope, that doesn't do anything. Okay. There I am. Any questions?
So we care about CoLE1 because it has this multi-copy system.
We care about CoLE1 because it's one of the few plasmids we've actually spent government money on to figure out what's going on. And so we use it as a conceptual model for how every plasmid must control its replication. Because it's a big question. Now, how does it do it? I mean, so how does something control its copy number if it's low copy? If it's 1 copy per cell, some just have 1 copy. It has an origin that works similar to ORAC. Probably involves recruitment of DNA A. Yeah. Let me go and look at an email I sent myself that contains some exam stuff, just to make sure I haven't forgotten any important things here. I mean, that's why you're really here, right? To hear me read the exam. But it's every year it's the same with me. It's not a mirror behind me, is it? Okay, yeah. I'm safe. Here we are. Okay.
Dr. Gurley, if you could, their critical thinking skills are on look. They want to know how a critical thinking question would be done for CoLE1 replication.
Oh, I just told them. Critical thinking might be done if you had a really evil instructor. He might ask you, how would you change copy number? How could you manipulate the system to increase or decrease the copy number that's naturally there? And so in general, if you want to increase copy number, you want to either mutate the promoter that makes the primer so it's more active, so you make more primer, or you want to make sure the negative components are not as abundant. You can mutate their promoters, so either you don't make as much RNA1, antisense RNA, or not as much ROM protein, so you don't facilitate formation of that duplex. So it's not difficult. I'm giving it away. So you're going to have a total of 8 questions. I'm giving you an hour. So if you still are there after an hour, you might want to hold a mirror in front of your nose and see whether you're breathing. There's nothing wrong with taking an hour. You're okay. I'm just being cruel. There'll be 4 questions on agrobacterium and 4 questions on all the rest. I can read the names. I can't read the names of all of my question banks, but I have 1, 2, 3 question banks regarding agrobacterium and 1, 2, 3, 4 question banks on plasmid compatibility, coli-1 replication, C plus D, that kind of stuff. I have the question banks on the Vir genes. So I'm going to randomly draw, and which Vir genes am I talking about? It's going to be Vir A, Vir G, that's receptor and effector. Those 2 are the only ones that are constitutively expressed all the time, right? Because you need to have the reception activation system there all the time to be alert to the signal. And everything else is induced by this activated Vir G, right? You're going to need to be able to compare the function of some of the Vir proteins with the proteins in TRA. Vir D1, Vir D2, that's like the TRA Y and I. I does the cutting in TRA, and Vir D2 does the cutting in the Vir region. You need to know what the wound signal is, acetylcyangone. You'll need to know what that secondary signal is, arabinose or some complex sugar like that. So it's actually a simple sugar. Arabinose. What does Vir A bind? Acetylcyangone, and it binds it in that transmembrane or just below transmembrane region, not in the periplasmic space. Where does ChEV interact? Well, when it binds arabinose, it interacts in the periplasmic domain. Need to know what overdrive is and which protein binds there. Overdrive is located at the right border of the tDNA. It ensures that the right side is cut first, and Vir C binds it. You need to know how a co-integrant is resolved in replication if you have a accidental recombination occurring. It's the Zer site-specific system acting at a diff site. That diff site's usually pretty close to the termination region. At least it's within about 30 kb. You're going to need to understand the concept of a helper plasmid. What is going to be on a helper plasmid? If you're talking about bacterial conjugation, it's going to be TRA on the helper, something that's functionally analogous to ORT on the mobilization plasmid. That has several names. It's called BOM or MOB. It is a functional ORT, a site where the TRA I and Y can cut. So how much of the tDNA, how much of the TI plasma? TI plasma is about 200,000 bases, 200 kb in size, huge plasma, usually only about 1%. How much of that thing actually ends up in the plant? Actually, one strand of the T region. Just like in conjugation, you actually only send over one strand of the plasmid that's being transferred. The second strand is duplicated or replicated in the recipient cell, whether it's a bacteria in conjugation or a plant cell. Which proteins in the Vir regulon are responsible for guiding the T strand to the nucleus? How do they know to go to the nucleus? Nuclear localization sequences, which are really simple. It's only, if you have 5 amino acids in a row and 3 of them are positive, it's probably going to be a nuclear localization sequence. 2 of these are localized on VIR-D2. 1 is on each copy of VIR-E2, but you have many copies of VIR-E on that strand. So there's signals on every protein that says, take me to the nucleus.
Is that why there was that one question on exam 1 where it was like, what does TRAL-Y through I not have? And the answer was nuclear localization.
Maybe, I don't remember that. That's a really good question.
It was like helicase capability,
Yeah.
ligase.
Was nuclear localization one of the choices?
It was the choice that was the right one. It was also
Yeah, so, yeah, good question. Like I said, there will be probably one fill-in, well, yeah. You have to fill in the blank on the VIR proteins. So know how to write VIR. And then D1, D2, C, A, G. We haven't talked about B much, have we? VIR B. That is involved in channel formation. So what would that be analogous to in TRA? VIR D. VIR D has connections to the T4 secretory channel. VIR B seems to perform a similar role, something involved with channel. Okay. Without just giving everybody 100, I think I've done all I can do here, but I'm happy to answer questions to you.
Dr. Gurley.
the ZER stuff.
The ZER system that is a site-specific recombinase.
Yes. Is that homologous or non-homologous, or does it not matter?
It's homologous because it's looking for diff, has to be lined up with diff, but it's site-specific homologous recombination.
So does that lead to variation in the two cells?
Not really, because they just were replicated. You know, they're very closely related to each other. They just gave birth to each other. Yeah.
Okay.
So.
The last thing you said about the T1 channel, you said for B, right?
B, as in Bravo.
Okay.
Bow wow.
You said it was connected to the TRA-D or TRA-D?
It's analogous to TRA-D, delta, in the TRA system, because they have a channel connection. Any questions from afar?
There was some worry about figures still, and I just told them to look at their homeworks, and I thought that would be the best route. What do you think?
If they have a specific question, I'm happy to answer. I just don't know what they're asking.
I think they're just worried, like, I guess there's some homework questions that have figures on
Yeah,
them.
well, I would definitely know the homework questions.
Okay.
And I might give you figures that I manipulated, like moving bomb sites all around the place or something like that. So, but it's not going to be, I will not ask you to identify the order of genes that are in TRA or the order of the genes in VIR. You might need to identify the components of COLI1 replication. I can't remember. Is that a homework? That could be a homework. I constructed a figure like that at one time. You could label what is RNA species 1, RNA species 2, what is ROM. Yes, not too difficult, but that's all I can remember about figures. It's not real figure rich. What was your question?
Oh, so only the tDNA single strand is transferred to the plant, right?
Right, just that bottom strand is called the T strand of the T region.
Okay, so, and then it starts making opine in the plant cell?
Right, it has promoters, which are control sequences for transcription, that function in the plant, a eukaryote. And that was my experimental question when I came to the University of Florida. We didn't know what a plant promoter looked like. And I was working with the Agrobacterium TI plasmid, mapping genes, and I wanted to sequence the promoters so Agrobacterium could tell me what a plant promoter looked like.
Okay. So they're making opine in the plant, but it doesn't have the opine catabolism part.
Right, okay. The plant is engineered to make the opine, and it then secretes the opine, or at least it's kind of leaks out, and it ends up in the soil after rain. And Agrobacterium in the soil now has the job of metabolizing it. And on the TI plasmid, conveniently enough, it contains all the genes it needs to break down the opines. Most other soil bacteria have no clue what to do with the opines.
Okay, thank you.
The first opine characterized was called octopine. It was named after octopenic acid. See, you got me going, I can't stop. What is octopenic acid? It's found in octopi. When they get tired, when they come back from the gym, their muscles ache, not from lactic acid, but from octopenic acid. Whew, and that's the last thing you need to know. Now you're ready for med school.
Dr. Gurley?
Yep.
We have a question related to the makeup exam.
Oh, man.
I know. How does it work related to the makeup exam? Is it going to be just 1A or 1B, or is it going to be combined?
You mean the optional final?
Yes.
That's a bridge I haven't built or crossed. I don't know. I have to see. What I'd like to do is do what's best for the student. So, see, if you just do exam 1B, it's not going to have much impact on your final grade. So probably I will combine it. I mean, as it is, exam 1 doesn't have quite the impact that the other exams have. So you're probably not going to be in a situation where you're going to need to retake exam 1B. You'll probably retake the whole thing. Otherwise, it's not going to do you any good. So if your lowest exam grade is 1B, that's kind of unfair, right? There are only 8 questions, and you really need to take one of these high contributing exams like 3 or 4 or 2. So I'll make it so you get a chance to redo the entire exam 1. So it'll have an impact on your final grade. Okay, I'm through with the formal review. If people want to form discussion groups, they're welcome to hang around a while or not. I'm available online to answer questions, but I think we've covered just about every possible thing today that could ever be on this exam. Only 8 questions, people. Okay, good luck. Oh, I have some cameras up here. If anybody would like to sign up for one, if you return it to me in class Tuesday, I'm happy to let them out again.
Dr. Gurley,
Yep.
all good things online. They appreciate the review. I'm going to go.
Okay.
All right, thanks.
Good luck to everybody.